morning student, we will continue competency number 6.11 and today's topic is biosynthesis of heme and its regulation. Heme is synthesized in almost all tissues in the body, but main organs for heme synthesis are liver and RBCs. In RBCs, heme is synthesized in normoblast stage and it is not synthesized in matured erythrocytes or matured RBCs. The heme synthesis pathway is partly cytoplasmic and partly mitochondrial. Now, steps of heme synthesis. First step is synthesis of delta aminoleucinic acid. In this step, succinyl-CoA and glycine combines to form delta aminoleucinic acid in mitochondria. This step is catalyzed by an enzyme called as delta aminoleucinic acid synthase or ALA synthase. This enzyme requires a coenzyme called as pyridoxal phosphate. This step is the rate limiting step of heme synthesis pathway, and ALA synthase is the rate limiting enzyme of this pathway. The coenzyme is pyridoxal phosphate. This pyridoxal phosphate is synthesized in our body from a vitamin called as vitamin B6 or pyridoxine. Now, after synthesis of delta aminoleucinic acid in mitochondria, it enters into the cytoplasm, and remaining steps of heme synthesis pathway, not remaining, few steps of heme synthesis pathway occurs in cytoplasm. Now, this step, as I have told you, it is a rate limiting step. It is also has some clinical importance. A case is given here. A patient suffering from tuberculosis and treated with anti-tuberculous drug INH, isoniazid. A few months later, this patient develops anemia which is not responding to iron therapy. This is the case given, and the question is, explain the biochemical basis of anemia and how will you treat anemia. First, I will explain the biochemical basis of anemia. Now see, in the last slide I have shown you that the pyridoxal phosphate is the coenzyme required for the first step of heme synthesis. Now this pyridoxal phosphate is obtained from vitamin B6, that is pyridoxine. This pyridoxine, with the help of ATP, is converted into pyridoxal phosphate by an enzyme pyridoxal kinase in the cell. Pyridoxine is present in is a vitamin present in the diet. In the cell, this pyridoxine is converted into coenzyme pyridoxal phosphate by enzyme pyridoxal kinase. Now this INH drug is a competitive inhibitor of the enzyme pyridoxal kinase. This INH inhibits the enzyme, and hence pyridoxine is not converted into pyridoxal phosphate. When coenzyme is not available, the first step or the rate of first step is decrease, and hence the heme synthesis pathway occurs at slow rate. The same I have shown you here diagrammatically. The left side shows the enzyme pyridoxal kinase. Now see, pyridoxine and INH, they have structural similarity. Therefore, there is a competition between pyridoxine B6 and INH to bind at the enzyme. As the concentration of INH or inhibitor is more, this INH binds at the active site of the enzyme and does not allow this pyridoxine to bind on the enzyme. Therefore, pyridoxine is not converted into pyridoxal phosphate, and due to absence of this coenzyme, the rate limiting step of heme synthesis pathway is occurring at slow rate, and hence heme synthesis occurs in slow rate. Now, this is the biochemical basis of anemia. Now next question is, how will you treat this anemia? Because this patient is not responding to iron therapy, it is given in the question. as I have told you, there is a competitive inhibition in this step. Substrate and INH compete to bind at the active site of the enzyme. As the concentration of INH is more, INH binds at the active site. But if you remember, the competitive inhibition is a reversible type of inhibition. Therefore, if we increase the concentration of pyridoxine above the level of INH, then this pyridoxine displaces the INH from the active site and binds at the active site of the enzyme, and pyridoxal phosphate is synthesized. Therefore, we have to give high doses of pyridoxine or vitamin B6 intravenously to such patient so that the pyridoxal phosphate synthesis starts again. So here the anemia is due to deficiency of coenzyme pyridoxal phosphate. Some inhibitors of step 1 are cycloserine. It is a anti-tuberculosis drug again. It is an antagonist of pyridoxine B6 or vitamin B6. Glucose is also inhibitor of heme biosynthesis. It inhibits the enzyme delta aminoleucinic acid synthase or ALA synthase. Ethanol is also a inhibitor of heme synthesis pathway. Ethanol is converted into acetaldehyde, and this acetaldehyde again inactivates the coenzyme pyridoxal phosphate. So these are the inhibitors of heme synthesis. There are some stimulators of heme synthesis. The first stimulator is starvation, which stimulates heme synthesis because in starvation glucose is deficient. Then female hormones also stimulate heme synthesis or the enzyme ALA synthase. Drug like barbiturates, we will see later on how it stimulates heme synthesis, but it also acts as a stimulator of heme synthesis pathway. Now coming to the second step, as I have told you, ALA synthesized in the mitochondria enter into the cytoplasma. Now this step occurs in the cytoplasma. Two molecules of delta aminoleucinic acid condenses to form porphobilinogen. The enzyme is ALA dehydratase, which removes two water molecules from the substrate to form a porphobilinogen, which is a monopyrrole ring. Now this ALA dehydratase contains zinc and it is inhibited by lead. This is the monopyrrole ring seen in the hemoglobin structure. This is a porphobilinogen which is synthesized in the second step. The difference is here, if you see, amino group is extra, and at the same time you will have methylene bridges here, instead of which monopyrrole have methynyl bridge. Again, clinical importance of this step: small child suffering from anemia not responding to any treatment. On detailed history, it was found that he is playing with the toys made up of lead. Again, second case is a person working in a lead industry is suffered from anemia not responding to any treatment. Explain the biochemical basis of anemia. Just I have explained. In both the cases, there is lead toxicity. This lead inhibits the enzyme ALA dehydratase. This step is inhibited by lead, and hence second step does not occur and heme synthesis stops. Now third step, in this step 4 molecules of porphobilinogen combines to form a linear tetrapyrrole structure. Linear means they are arranged one by one from side to side to form a structure called as hydroxymethylbilane. In this process, 4 ammonia groups are removed from the porphobilinogen. The enzyme is porphobilinogen deaminase. This step also occurs in cytoplasma. The another name of PBG deaminase is uroporphyrin 1 synthase or hydroxymethylbilane synthase. This is the linear structure of hydroxymethylbilane, which is joined by methylene bridge CH2. But in heme, methynyl CH bridges are present. Now one more thing, these porphyrinogens are colorless compounds, but they readily oxidize to porphyrins, which are colored compounds. Now hydroxymethylbilane, which is a linear pyrrole structure, undergoes spontaneous cyclization to form uroporphyrinogen 1 in cytoplasma. That means the linear pyrrole structure now suddenly becomes a cyclic structure. So uroporphyrinogen 1 is the first porphyrin of heme synthesis pathway. Now this uroporphyrinogen 1 then converted into uroporphyrinogen 3 with the help of enzyme uroporphyrinogen 3 synthase in cytoplasma. Now what is the difference between uroporphyrinogen 1 and uroporphyrinogen 3? It is the difference at 7th and 8th position of 4th pyrrole ring. In uroporphyrinogen 1, there is acetyl group at 7th position and propionyl group at 8th position, whereas in uroporphyrinogen 3, propionyl group is present at 7th position and acetyl group is present at 8th position. In this way, uroporphyrinogen 3 is formed, but small amount of uroporphyrinogen 1 normally get converted into coproporphyrinogen 1 with the help of enzyme uroporphyrinogen decarboxylase. As I have already told you that porphyrinogens are colorless compounds. They readily oxidize to porphyrins, which are colored compounds. In fact, all porphyrinogens like uroporphyrinogen, coproporphyrinogen, protoporphyrinogens, they are colorless compounds, but on oxidation they form corresponding porphyrins, namely uroporphyrins, coproporphyrins and protoporphyrins, which are colored compounds. Now in the last step we have seen uroporphyrinogen 3 is formed. Now this uroporphyrinogen 3 undergoes decarboxylation to form coproporphyrinogen 3 with the help of enzyme uroporphyrinogen decarboxylase. It occurs in cytoplasma where 4 carbon dioxide molecules are removed. Here the acetate group, which is present in uroporphyrinogen 3, is decarboxylated to form methyl groups. So 4 acetyl groups on decarboxylation forms 4 methyl groups, and the compound is now called as coproporphyrinogen 3. Now this coproporphyrinogen 3 again enters into the mitochondria and further reaction occurs in mitochondria. After entering into the mitochondria, coproporphyrinogen 3 undergoes oxidative decarboxylation to form protoporphyrinogen 3 with the help of enzyme coproporphyrinogen oxidase. Here oxidation occurs by removal of 2 hydrogen atoms which are accepted by NADPH, and decarboxylation occurs by removal of carbon dioxide molecule. Here the 2 propionic group or propionic acid group which are present in coproporphyrinogen 3 are converted into vinyl group by loss of carbon dioxide molecule. Here molecular oxygen is required. This molecular oxygen is converted into water and the hydrogens are donated again by coproporphyrinogen 3. In the next step, protoporphyrinogen 3 is converted into protoporphyrin 3. Protoporphyrinogen is converted to protoporphyrin. So this step occurs in mitochondria. Here oxidation takes place. The enzyme is protoporphyrinogen oxidase. Again oxygen molecule is required. 2 hydrogen atoms are removed from methylene bridges which are get oxidized to methynyl bridges and these two hydrogen atoms, or these hydrogen atoms, are donated to oxygen, which get converted into water molecule. Now this protoporphyrin is a colored compound. Now this protoporphyrin 3 combines with ferrous iron in mitochondria in the presence of enzyme heme synthase to form heme. The heme synthase is also called as ferrochelatase. The iron atom linked with 4 nitrogen atoms of pyrrole rings to form a structure of heme. This is the complete heme synthesis pathway shown here. Now we will see regulation of heme synthesis. As I have told you, ALA synthase is the rate limiting enzyme of this pathway. So this enzyme is regulated by different ways to regulate the heme synthesis pathway. The first method is repression mechanism. In this diagram, I have shown you genes which synthesize the enzyme ALA synthase. So first is, on the left side, I have shown you regulatory gene or regulatory DNA. This DNA on transcription translation forms a protein called as aporepressor. This aporepressor is a dimer. Dimer means it is made up of 2 subunits. Now on the right side, I have shown you promoter region where RNA polymerase binds, then operator region, and next is structural gene responsible for synthesis of the enzyme ALA synthase. When this gene is translated, the heme is ALA synthase is synthesized, and this ALA synthase then stimulate the process of heme synthesis. So this, on the right side, I have shown you the heme which is synthesized by the enzyme ALA synthase in a small 2 square boxes. Now what happens here? The regulatory gene synthesize aporeceptor, which are shown by 2 circles. These 2 aporeceptors or dimer aporeceptor is inactive. When heme synthesis is there inside the cell, or when heme is available in large quantity in the cell, at that time this heme binds with the aporepressor. This heme is called as co-repressor. Now this aporepressor and co-repressor combines to form a tetramer called as holorepressor. Now this tetramer holoreceptor repressor is active, which binds at the operator site. Due to binding of holorepressor at operator site, RNA polymerase, which is present at the promoter site, cannot move to the structural gene, that is ALA synthase. Therefore, there is no transcription of ALA synthase gene, and when there is no transcription means automatically the concentration of ALA synthase decreases, which in turn decreases the heme synthesis in the cell. So in this way, the heme inhibits its own synthesis by repression mechanism. Here you have to remember, the regulatory gene synthesize a aporepressor, which is a dimer, and dimer is inactive. But when the heme is available, 2 molecules of heme binds with the aporepressor and forms a tetramer. Now this tetramer is active, and then it binds with the operator site and stops the heme synthesis pathway. The enzyme ALA synthase is also allosterically inhibited by hematin. Hematin means when heme is present freely without globin chain, then ferrous atom which is present in the heme is converted to ferric form, then that heme is called as hematin. Now when there is large quantity of heme is synthesized. Therefore, heme is available in free form and it get readily converted into hematine. Now this hematine binds at the allosteric site of enzyme ala synthase, because ala synthase is a allosteric enzyme which has allosteric site for binding of hematine. Another site is active site where the substrate succinyl CoA binds. But due to binding of hematine at the allosteric site, the hematine brings the conformational change in the structure of allosteric site, and hence the substrate does not binds at the active site of the enzyme and heme synthesis stops. In our body, ala synthase is having two forms. Hepatic form, also called as ala synthase 1, erythroid form is called as ala synthase 2. Hepatic form is induced. Induced means increased synthesis. Hepatic form of ala synthase is induced by the drug like barbiturate and female sex hormones, and this hepatic form is also subjected to inhibition by heme. Whereas this erythroid form, also called as ala synthase 2, it is not induced by any drug and it is not subjected to feedback inhibition by heme. So this is the difference between the ala synthase of hepatic form and erythroid form. Next regulation is by feedback inhibition. Just we have seen uroporphyrogen 3 is converted to coproporphyrogen 3. Coproporphyrogen 3 is converted to protoporphyrogen 3. Whenever the concentration of product increases, it inhibits the enzyme by negative feedback mechanism and stops its own synthesis. For example, when uroporphyrogen 3 is converted into coproporphyrogen 3, this coproporphyrogen 3 inhibit the enzyme which synthesizes or which converts the uroporphyrogen 3 into coproporphyrogen 3, and in this way the concentration of coproporphyrogen 3 decreases. Similarly, when the concentration of protoporphyrogen 3 increases, it acts on the enzyme which convert coproporphyrogen 3 to protoporphyrogen 3, and the synthesis of protoporphyrogen 3 stops and its concentration decreases. But this type of inhibition is not seen in series 1 protoporphyrogens or porphyrogens. Uroporphyrogen 1 is converted to coproporphyrogen 1, coproporphyrogen 1 is converted to protoporphyrogen 1. But this type of inhibition is not seen in series 1. Normally, the concentration of series 1 porphyrogens are less. Series 3 porphyrogens are dominant in our body. It is shown here. I have shown you that uroporphyrogen 1 is normally converted into uroporphyrogen 3 in large quantity, but small amount of uroporphyrogen 3 is converted into coproporphyrogen 1, which is then converted to protoporphyrogen 1. Next, regulation of heme synthesis is compartmentalization. Compartmentalization means different enzymes of different steps of heme synthesis are present in two different compartments. Some are present in mitochondria and some are present in cytoplasma. For example, the enzymes of step 1, 5, 6, 7 are present in mitochondria, whereas the enzymes of step 2, 3 and 4 are present in cytoplasma. Next is drugs like barbiturate and anticonvulsants drugs. Anticonvulsant means epileptic drugs, anti-epileptic drugs. Now these drugs require heme containing cytochrome P450 for their metabolism, or you can say the barbiturate and anticonvulsant drugs are removed from the body with the help of cytochrome P450 present in the lungs. Now this cytochrome P450 synthesis require heme. Therefore, when we consume these drugs, the synthesis of cytochrome P450 increases, which will utilize the heme, and therefore there is depletion of heme. And when there is depletion of heme, the feedback inhibition of ALA synthase by heme is removed, and there is induction of heme synthesis, or there is increase in the heme synthesis. Out of total heme synthesized in our body, 2/3 of heme are utilized for synthesis of cytochrome P450. So cytochrome P450 is a important enzyme system required for metabolism of various drugs in our body, like barbiturates and anticonvulsants. This we have already studied the mechanism. The step catalyzed by furochelatase or and aldehydatase is inhibited by lead. The last step and the second step is inhibited by lead. So in lead poisoning, again heme synthesis is decreased. INH, I have told you, INH decreases the availability of pyridoxal phosphate and affect the heme synthesis, that we already studied. Glucose in high concentration prevents the heme synthesis by inhibiting the synthesis of the enzyme ALA synthase. So this is about the regulation of this is about the heme synthesis and its regulation. Thank you very much.